So our next speaker is Raluca Matescu, who is down at the University of Florida. I've had the honor of working with Raluca for probably a better portion of a decade on a number of different projects, and she'll talk a little bit about things in which she's doing with heat stress in beef cattle.
Great. Thank you. Thanks, Jim. Thanks for the opportunity to be here. Really excited to hear all these talks and hopefully try to figure something out by the end of the 2 days. So this is the title that I gave to Jim before I realized I only have half an hour. So, but we'll talk about some challenges and some opportunities. And I wanted to start by looking at some of the areas, and these are research areas mainly in beef cattle, where I think that, you know, the high throughput phenotyping is going to be very beneficial, and where moving into the phenomics is going to have the highest impact. And you definitely have the first one, that's an area where we are making progress. There's a lot of research going on, and there's a lot of effort that's being put into production efficiency traits, feed intake, reproductive performance, survival. But then you have other research areas which are very important, and they are important for the producers and also for the consumers and the public. And then if you look at those, so adaptability is probably at the top of the list, with heat stress tolerance probably the best example going in there. Improved animal health and welfare, disease resistance, it's a very good example. But also enhancing the healthfulness of animal products for human consumption. So we talk about fatty acid composition or mineral content. And then you get into something like environmental impact, the air, water, soil quality, and also the impact of wildlife. So if you look at those type of traits, you know, the challenge there is really that right now we don't really have, it's our inability to measure those traits easily, cost effectively and reproducibly. So, and of course on a large number of animals. So to my mind, you know, these are the traits that are really going to benefit the most, and where if we can find a way to implement the high throughput phenotyping and get into phenomics, will have the biggest impact in terms of improving the performance and the efficiency of livestock systems. So what are some of the challenges for advancing the field? They are related to our, as I said, our inability to measure those traits frequently enough. So if you think about the traits I had on the previous slide, most of them are not static, they are dynamic. So they really change through time, they change over different environments. So I can't really take only one measurement of that one particular trait. So we really need to describe accurately that phenotype, you really need high resolution and multidimensional data. You need to do it through time, so different developmental stages across multiple environments, and also you need to look at them at different multiple levels, cellular levels, tissue level, organ levels, and we heard that from Natalia this morning as well. So in terms of key concepts, when I think about it, or at least key steps into trying to move into phenomics, there are really 3 big steps we need to make. The first one is developing the methodology to measure the traits, and that's what I'm going to focus on today, and I'll show you some results or some of the things we do with heat stress in cattle. The second step is, once we figure out what we need to measure and how we need to measure it, we need to develop, either find the equipment or develop the equipment that we need to acquire the type of data that we need. And then, of course, we need the computational approaches to be able to integrate all the data and analyze it in some manner. So I'm going to focus on the first step, which is developing the methodology to measure the trait, and I'm going to illustrate that in the context of some of the work that we are doing, which is part of a USDA NIFA grant. It's breeding climate-smart beef cattle. And the overall goal is to increase tolerance to heat stress, but we want to do that by allowing for, in the same time, for increased efficiency of production, reproduction, and meat quality. So, thermotolerance. Thermotolerance is one of those traits, just like disease resistance, where the challenge is how do I identify the individuals which are tolerant to heat stress. And to do that, you need to expose your population to that stressor. So in this case, you need to expose your population to the heat stress and identify which are the animals which are tolerant. Now, I am in Florida, so we have that opportunity to expose our animals to heat stress every single summer, and the project is going to look at about 2,000 Brangus heifers. These are 2-year-old heifers, and we are going to try to measure a number of phenotypes on them. The first phenotype, which is really a measure of the thermotolerance or body temperature, is the vaginal temperature. We measure this with some with an eye-button temperature logger, and we do it over 5 days. So we end up with continuous measurements of vaginal temperature over 5 days, every 15 minutes. We actually have 3 days because the first day is when we insert the seeder, the last day is when we take it out. So we really have 3 days of continuous data or measurements of the vaginal temperature when these heifers are on the pasture and they are undisturbed. And then we have the environmental data, which is taken in the same time, still every 15 minutes, temperature, humidity, so we can compute the temperature humidity index. We are trying to measure sweating rate, and I'm going to show you this and some of the challenges related to sweating rate. But this is an important phenotype for us because it will we think about it as a component trait actually of the thermotolerance. Coat, we are looking at coat color, coat score, hair length, and hair diameter. We are recording temperament, the shoot and the exit score, body condition score. We are taking 2 skin biopsies on each one of these animals. One we are using for histology work, and one hopefully for some transcriptomic work at some point if we can get the funding to do it. And we are also going to have weight gain over the summer and fall and subsequent pregnancy status on all these animals. So these are all replacement heifers, so they are going to go through a timed AI. Actually, we are getting ready to do that in December, so we'll have the pregnancy status on them. And the last two, the weight gain and the pregnancy status, we hope that we can use the data to identify, because the goal is to identify not only individuals which are heat tolerant, but we want to identify those animals which are able to maintain their body temperature in a normal limit, not by decreasing the feed intake or penalizing some of the other production traits. We want those animals that are able to do it through other mechanisms. So we are going to try to do that by using the other traits. So I'm going to show you some of the data, and I'm going to show you some of the challenges and also some of the opportunities. And these are 4 plots. So this is work that we've done over the summer. We had about 800 heifers. We did it in groups of 200. So we had 4 groups. Each group is 200 heifers. And what you have in this graph is just the THI, so the temperature humidity index for those 4 groups of cattle. For each group, as I said, you have 3. No, I'm gonna use Jim's fancy pointer. All right, so, oops, but I went too far. Okay. Sorry about that. So for each group you have 3 days, so you can see the THI over those 3 days for the 4 groups of cattle. So the first thing, remember, the goal is here to expose those animals to heat stress and then measure their response, okay? So when we first look at those 4 groups, we said group 4 is the ideal group. So here I have 3 days which are almost identical. THI goes pretty high and it's pretty uniform and it's high throughout the day. When you look at the other 3 groups of animals, the THI during those 3 days, it's pretty variable. So first of all, each day it's kind of different within a group, and then even within a day, if you look at group 2, the red line, the THI goes up in the morning, but then it kind of drops down. So even within a day, I have a different level of the THI. So we thought that this is going to be a problem. But then when we looked at it a little bit closer, we thought that, that gives us an opportunity to use this type of data and look at some of the differences. Also remember that within each group, what I have exactly the same animals. So these are 200 animals which are exposed to different level of THI every single day. And so what we did, we took two groups where I do have, within each group, I have 2 days which are very different. So I'm going to only show you those 2 days. So I have a day in which the THI is pretty high in both days, and then I have another day within the same group where the THI is not that high. So that allows me to make some comparisons. I can look at the same animals and see what is the response of those animals in a day with a high THI versus the response under a lower THI. So when we look at that, so we combine those two groups, and this gives you an overall image of the difference in THI between a low heat stress versus a high heat stress day. So you have about a difference of about maybe 5 units of THI between those two. And then when you look at here what I have is the vaginal temperature of those cows over the 2 days. So you can see that so I have exactly the same cows, their vaginal temperature is going to be lower in a day when I have a low heat stress situation, but then it's going to be higher under a high heat stress situation. But this allows me to calculate for each level of THI what is the increase that I expect to get in that vaginal temperature. What I also want you to notice in this graph, which is very important for the type of work we are trying to get done, is the amount of variation you have in that vaginal temperature. So for every single point, right, I have, and remember, these are all Brangus heifers, they are all 2-year-olds, they are on the same, you're looking at exactly the same environmental conditions, but I do have a tremendous variation in terms of their vaginal temperature, which for us, it's pretty good. We looked at the code score, and I hope you can see it. So I'm going to show you only the difference between a code score of 1 and code score of 2. Score 1 is what we call excessively smooth, score 2 is fairly smooth, so it's a little bit longer, and I think that you can see the difference. When you look at the code score, it's actually, oops, I'm going the wrong way. It's actually nice to see that we can see that effect of the code score on the vaginal temperature. So what you have here in blue, those are the cows that have a code score of 1, so very sleek hair. And then in red you have cows that have slightly longer hair. And you can see the difference. This is a significant difference. This is over all the groups and over all 3 days we had, so about 800 cattle in total, and you can see a very nice difference, which is statistically significant, which is due to the hair. But then we thought, well, how about if I just look at that effect of the code score, but now I'm going to use my 2 days, which are very different. So what is the effect of the code score if I look at a high heat versus a low heat stress situation? And I'm going to show you actually only to make it a little bit easier to look at, I'm going to show you only 4 hours. So I picked the time of the day when I have the highest difference between THI between the 2 days, and these are my 4 hours. So I'm going to show you data for only 4 hours throughout the day. And what you have for each hour, you have red and blue. So the blue are the cows under a low heat stress situation, and then in red you have exactly the same cows but under a high heat stress situation. And then the color, lightness of the color, so the light blue, it's a code score of 1, and the dark blue, it's a code score of 2. So the shade of the color is really the code score, with the darker color showing or being associated with a longer code. And there are a number of things that I want you to see on this graph. So first of all, you can see an effect of the code score even under a low heat stress situation. So those animals that have a longer code are going to have a higher body temperature. But you can see that that effect, it's almost double, if not more, under a high heat stress situation. Okay, so we can calculate that. But the other thing which I think is very nice in this graph is the amount of variation under the two different days. Okay, so remember that again, I have exactly the same cows in blue and in red. Under a low heat stress situation, I don't have that much variation in that internal temperature, while under a high heat stress situation, I've got a lot more variation to work with, which makes sense, and it also underlines the score, underscores the point that, you know, we need to take those measurements under the right conditions. So I need to be able to expose those animals to that high stress if I'm going to be able to identify the animals which are the ones which are tolerant. So not every measurement under every condition is important. We really need to get those measured under the right conditions. Sweating rate, it's a very challenging measurement for us. We can technically do it. We cannot do it remotely, so we need to do it when the animals are in the chute. So we did this when we actually put the eye buttons in the cows. And so you are going to see a time effect. Okay, so, and you can see that, so these are 3 groups of cattle. You've got the sweating rate. In them, you definitely see the time effect. The first cows which are coming through, first of all, it's really early in the morning. It's not hot, it's not humid. They're the first ones to come through. They are not sweating that much. Later in the morning, it's getting hot and it's getting humid, and they've been also waiting in the corral to come through the chute, so they are going to be sweating a lot more. So although we can definitely account for the time effect, again, the ones here in the morning, they are not exposed to the same level of stress as the ones later in the day. So that's going to be a problem in terms of the accuracy of that measurement and what we want to get out of it. But I think it's also nice to, if you look at this graph, even though you have a time effect, if you take any time point on this graph, you can still see a large amount of variation. So there's still quite a bit of variation in that sweating rate, even within, you know, at any given point in time. So sweating rate, as I said, that's an important, what we think about it as a component trait. So I want to make sure maybe that's what I want to find. I want to find those animals which are sweating more, so they are able to dissipate a little bit more heat through sweating and not by decreasing their feed intake or any other physiological traits. So it is important to be able to measure it. We just need to find a way to measure it a little bit better. And in an effort to actually try to get to that sweating ability a little bit better, what we are also doing, we are taking skin biopsies. And as I said, one of them, we are using it for histology. And this is a picture, a histology picture on one of our cows. What we hope to be able to do with this histology is, first of all, we can measure the depth of the dermis and epidermis. We can measure the number of hairs and the number of sweat glands. We can also measure the sweat gland depth, and we still need to figure out, but we hope that we can measure the sweat gland volume. So hopefully this is going to give us a little bit more detailed phenotype that we can correlate with the sweating ability. So I'm going to conclude by saying, you know, any trait is going to definitely benefit from high throughput phenotyping. There's no question about it. But I think that high throughput phenotyping is going to be essential for the traits where we have a difficulty today of measuring. So that's where the highest impact is going to be in terms of figuring out how to do it. But it is a process, okay? So it starts, and just like I showed you with our trait, it starts with developing the methodology, developing and refining the methodology to measure the target trait. We need to think about the traits that we are interested in and figure out, and this we already heard this morning, can I deconstruct that complex phenotype maybe in some other component traits? Are those component traits maybe a little bit easier to measure, or maybe they have a higher heritability? So there are a lot of questions. And then of course for each one, you're starting again. When should I measure it? How should I measure it? And things like that. There's always a question which way to go. And of course, there's some balancing you need to do. Are you going to go through a systematic phenotyping, measuring a few traits on many animals, or are you going to try to do many traits on a few animals? We try to do a little bit of both. So we try to do as many traits as we can on a large number of animals. But then that's really just the first step. So once you have the methodology and you kind of know what you need to measure, you need to move into how do I actually measure that on a high throughput and broad scale. And broad scale. So that's just the start of the process. And we heard this, it goes without saying, you know, we've been talking about interdisciplinary research for a long time. I think we need to take that and multiply it at least by 10, but you need a lot of different people in lots of different fields if we can make this work. So I'm going to stop there, and if there are any questions, I'll be happy to answer.
So the question I have comes about if you want to make animals more productive in stressful environments, should you be trying to select for adaptation, or should you be trying to select for performance in that environment? And my argument would be that if you selected these animals so that they grew efficiently and they reproduced efficiently and produced a quality meat product in that environment, you'd get the adaptation you were looking for.
Yes, and that's true. I think that one of the problems, you know, that you have with beef cattle is that they are all over. So even though if you take Florida, right, we do have most of our producers are cow-calf producers, but they are going to send those animals somewhere else for finishing. So they might be interested. So, you know, you talk about adaptability for Florida for a cow-calf producer, but they still need to sell their product, and that product is going to go somewhere else. I think that, you know, what really we want to do is, and that's going to be the challenge, I want to really understand what are the genes that are driving that adaptability, particularly to the heat stress, so maybe, and disentangle them from the production trait. So then regardless of where they are, I can still maybe select or try to integrate those genes into other breeds without bringing all the other production traits that I have with them.
So I have two questions. The first is, your assumption is that the animals that have a lower body temp are better able to handle heat, right? Perhaps an animal is heat stressed at different thresholds on body temp. So I don't know that selecting for the lowest temp is always the right idea. I guess I would like you to maybe address that a bit. And then the second one is, how did you decide that 3 or 5 days, whatever you're doing, is enough to get good data, as opposed to maybe 1 week every month during the summer?
Yes. So I'm going to start with the second one because it's easier. It's always about, the resources that you have and the things that you can do with the money that you have. So the iButtons that we are using, they are good for about, and they are not cheap. They are about $75 a piece. So we can collect data at 15 minutes interval for about 15, 20 days. So we wanted to use the same iButtons on all the groups of animals that we have. So we are recycling them and go through the groups of animals. So we can really do it for only about 5 days. If we can have more data, I think that, it depends on what type of days you also have. So as long as you're looking at that high THI and you have a good, good enough days, I think the data is pretty consistent over time.
Did you look at repeatability across?
We did look a little bit, but not too much. But I think, if we use that last set where all 3 days are almost identical, and that gives us a little bit of an indication of how repeatable those measures are. Yes, we can definitely use more days and you'll get more data, but I think to start, I think 3 days is enough. The first question was related to, could you repeat your question?
So the assumption is that the animal with the lower body temp is desirable.
Yeah, we are really not looking for the one with the lower. We are looking for the one that it's able to maintain that temperature within normal limits under a high heat stress situation. So, we are looking for about, if it goes over 39.5, that's when we are worried about, because that's where we know that you're going to start seeing some effects on reproduction and other traits. So I'm looking for an animal that's able to maintain that lower body temperature throughout the heat stress.
But you're assuming 39.5 is based on the average animal, right? So I think this gets back to Jerry's question that we don't really know for sure what temperature is desirable, I think.
No, and maybe that's where, if we look at our different days, that's maybe the answer, because I can look at what's the vaginal temperature on this cow under a low heat stress situation, and then see what's going to happen, how much higher that vaginal temperature gets when it's really hot, and then look at the difference and maybe select them based on that. So even though regularly they have a little bit higher temperature, even in a low stress situation, we are really looking for the difference and how much they are going to increase that vaginal temperature.
I have a question about the technology that you used. I know these iButtons only collect data, they don't transmit it, but hypothetically, if you could transmit it, do you think it would be useful to manage animals, or that there wouldn't be enough value, say, to try to prevent heat stress before it happens?
That's a good question. If you ask me, I think there is the raw data, it's very noisy. So, we have producers that are trying to use this data to know when the animals are going to calf. And they get those signals on their iPhones, and if you look at the data, the raw data, they would be there every single second, because you're going to get random spikes in those temperatures, which doesn't really mean something. Now, it could probably be cleaned up, and the whole process could be, with some data analytics you can do, but I think right now you are not going to use this type of real-time data for management. I think you're going to use it for, trying to improve the trait more. Yes.
Hi. Yes. Do you have the opportunity to look at your 39.5 threshold and do a straightforward variance covariance analysis with pedigree animals and look at the heritability and genetic correlations with performance traits? Because that will tell you whether it's a new phenotype which you could use for a genetic evaluation.
Yeah, we are just starting this work. So, but yes, we hope to do that. So we do have the pedigree on these animals. We are genotyping everything with the 250K, which is the latest functional chip, which works very well on this population. So yes, we hope to do that.
So I'm going to ask one last question
Yes.
because I think your answers to some of the questions actually sets it up. Is not, in reality, to some extent, the problem a lack of technology that you have to go back to the victors of the world and say, we need something better that costs a heck of a lot less than $75 a button in order to
Definitely.
do what it is that you need to do?
Yes, definitely, you know, better technology and cheaper would definitely help. But I think that what you have right now, we can look at least try to figure out what exactly is it important or critical that we measure. And then once we kind of know this is really what I need, then I think, but definitely bringing the technology and making it more affordable definitely will help with getting more phenotypes.
Well, if you'll join me in thanking Raluca for a wonderful talk.